The structures page can be found in the main GPCRdb menu under the structures section here. It takes a few seconds to load in because of the amount of data it stores. We also refer to this page as the structure browser because that is its main purpose. You can browse structural data in this table format. Every row in this table represents a structure, and you can find all sort of structural properties in the columns. We divide this data into several sections, and you can scroll to the right to view all of them. But if you're not interested in some of these and you want to make the table more compact, you can just hide sections by clicking on the X on the right-hand side of that. Also takes a few seconds to hide these sections, but you can see now that is removed. But if you want to show the hidden columns again, you can just press the Show Hidden Columns button here at the top. And in a few seconds, it will show the hidden columns again, or you can just refresh the page. The next button I would like to tell you about is the representative structures state and receptor. So by default, you can see that we have now 793 entries in this table. That is all the structures that are available in the database right now. And what you can see is that there might be multiple structures for the same receptor in the same activation state in this table. But if you would like to just pick one that might be sort of a representative structure for that receptor in that activation state, you can just click on this filter, and it will filter the whole table for you. So you can see now we only have 195 entries. So for one receptor, this picks several representative in the sense that one per activation state. So for the beta 2 receptor, there would be one representative structure in the active state and one representative structure in the inactive state. If you want to show all structures again, you can just press the button again, and then it will remove the filter. What you can also do here is that if after you're selecting some of the entries like this, clicking on the row or clicking on the checkbox here, you can press the Align Sequences button. This will redirect you to the sequence alignment page. You can also download these structures in the PDB format by pressing this button. Furthermore, you can make structural superpositions by pressing this button. This will show this pop-up page where you can select which one from the selected should be your reference structure on which the others are superpositioned now. After selecting that one, it will redirect you to the superposition page. You can also download the table in an Excel format by pressing this button. Let's go through some of the data columns. So the first section here on the left is the receptor section. You can find links to the UniProt page, the receptor, link to the GPCRdb page of that receptor. I'll show you. So it opens up a new tab. Here you can find the sequence, snake block, and so on of that receptor. Also receptor family, class, and species. So you can filter all of these here on the top. Just so you know that these are multi-select filters, so you can select multiple at the same time, like this. And also, I want to show you this little icon here. So if you select a couple of entries like this, then you press this icon, you can export the UniProt IDs. or the UniProt entry names in other Word, and that copies those entry names to the clipboard. So you can use that for further analysis on other GPCRdb pages. The next section is the structure section, where we store some details about the experimental structure itself. So for example, experimental method, PDB code, which has a link to the structure page, where you can actually view the structure in 3D. Then also a link to the refined structure if it's available. We provide these refined structures where we fill in missing coordinates and revert mutations to wild type. Also resolution, a preferred chain. If there are multiple chains of the receptor in that same structure, we pick one with the highest sequence coverage. Then activation state, and with the degree active column here, where 100% resembles the fully active structure bound to G protein or arrestin, and the 0% is fully inactive, and the structures in between are calculated on a residue distance relative to these two endpoints. But more on that, you can find information here. And also if you follow the documentation links here. Percent of sequence, so that's just the sequence coverage. Then other sections, signaling protein, if it is in the structure. We also have a link to the protein page of that signaling protein. Then some other sections, auxiliary proteins. So if there's a fusion protein in the structure, or antibodies, nanobodies. Structure ligand. Here we have links to the GPCRdb page of the ligand, where you can find all sorts of information about that specific ligand. And then the type of ligand and the modality as well. Then the endogenous ligand, sodium ion site, whether based on the conserved residue positions that make it possible to bind sodium, whether that's in the receptor itself and whether the sodium ion is actually in the structure. Then the reference section, here you can hover over the author's name to expand the full author list, and also a link to the actual paper, when the structure was published, and the annotated column just resembles the fact whether this structure was manually annotated when it was put into our database.